In this lesson, we're going to talk about hemoglobin and oxygen dissociation curves, and here are your objectives. Now, before we get into that, we should try and understand the concept of the partial pressure of a gas. Now, you may know that the air pressure at sea level is about 700 millimeters of mercury, and oxygen is about 21% of that. So if we take 21% of 760 millimeters of mercury, we get about 160 millimeters of mercury. Now that is the fraction of this total air pressure that is provided by the oxygen, and so we call that the partial pressure of oxygen, or the PO2. This is important because oxygen and other gases move from higher partial pressures to lower partial pressures. That's how they diffuse. So it's kind of analogous to concentration. Now it's not the same as concentration though, and that's actually important. If we have air here that has a partial pressure of oxygen of 160 millimeters of mercury, and then we have some water underneath which has no oxygen in it at all, so partial pressure of 0. If we let this come to equilibrium, over time some of that oxygen will diffuse into the water, and it's diffusing from an area of high partial pressure to an area of low partial pressure. And if that's being replenished from above, if the air is being replenished from above, so its partial pressure of oxygen stays the same, when you've let this come to equilibrium over a long time, you will end up with a partial pressure of oxygen in the water that is also 160 millimeters of mercury, because that diffusion will keep going until these two numbers are equal. Notice though that the concentrations are different. Oxygen has very low solubility in water, and so the concentration down here is low, but the partial pressures are the same. Again, we have the same partial pressures, but different concentrations. And in fact, it's because of this low solubility of oxygen in aqueous solutions that we need hemoglobin in the first place. If you had to carry oxygen in your blood just dissolved in the plasma, in the liquid part, you would need to pump a whole lot of blood in order to deliver enough oxygen to the tissues. But instead, hemoglobin serves as a kind of transport truck, and it can pick up oxygen and allows a lot more oxygen to be in the blood in solution, because it's carried on these carriers, not just dissolved in aqueous solution. So now let's look at the structure of hemoglobin and how it lets hemoglobin do that. Hemoglobin is a protein, and it is made of 4 polypeptide chains. It's a globular protein, or a sort of roundish protein, and so these are actually called globin peptides. And there are 2 different subunits, 2 different polypeptides making up the hemoglobin protein. 2 of them are called alpha and 2 of them are called beta. So you can imagine that you have an alpha subunit there, another alpha polypeptide there, and then 2 different amino acid chains, and these will be the beta chains. And each of those has a group on it called a heme group. That is a non-protein ring structure that has an iron in the middle, and it's the iron in the middle that binds the oxygen. So if this in yellow is the oxygen here, each hemoglobin molecule can carry 4 oxygens, 1 on each iron on the heme group. And because of the structure of these 4 subunits, and because of the way they interact, hemoglobin displays an interesting property called cooperativity. I should have mentioned, by the way, that this abbreviation HB is often used for hemoglobin. So each oxygen that binds to hemoglobin changes the shape of the hemoglobin overall, and makes it easier for the next oxygen to bind. So the first oxygen that jumps on makes it easier for the second oxygen to jump on by subtly changing the shape of the hemoglobin. And that makes it easier again for the third to jump on and for the fourth to jump on. And vice versa as well. The first oxygen to jump off makes it easier for the next one to jump off, etc., etc. And this cooperativity promotes oxygen loading at the lungs, because once the first oxygen is on, it's easier for the next ones to get on. And it also promotes oxygen unloading at the tissues, the muscles, etc., that are performing cellular respiration and need oxygen in the first place. So cooperativity promotes oxygen loading at the lungs and unloading at the tissues. Now let's look at what are called oxygen dissociation curves. And these are graphs that have partial pressure of oxygen on the x-axis and the resulting saturation of hemoglobin, or how full hemoglobin is, on the y-axis. So what this is telling us is that the different partial pressures, like the partial pressures of oxygen that we might find at the lungs, and let's say at the tissues, like resting muscle, how full is the hemoglobin? Well, it turns out that hemoglobin is almost fully saturated, or almost completely full, carrying as much oxygen as it can at the lungs. Not quite 100% saturated, but close. So if that's 100% saturation, the hemoglobin saturation at the lungs is a little bit below. And at the tissues, it's lower. So let's say that's about 70% at resting tissues. And if you trace this out for all the different partial pressures, it turns out that the graph looks like a kind of S shape like this, or has a sigmoid shape. And that is a result in part of the cooperativity that we talked about before. So hemoglobin is almost fully saturated at the lungs, and it releases oxygen at the tissues. Now, this might be the partial pressure here of resting tissues. The partial pressure of active tissues, like exercising muscle, is even lower. And this gap that we see here between 100% saturation and 70% saturation reflects the oxygen that has been unloaded from the hemoglobin. If at the partial pressure of the lungs, the hemoglobin is almost 100% saturated, and at the tissues it's 70% saturated, this gap reflects the oxygen that's been unloaded. It was picked up at the lungs, but then has been offloaded at the tissues. And here, if this is the partial pressure of more active tissues, then now we can see that even more oxygen has been unloaded. So that's kind of what these oxygen dissociation curves are describing. Now, it turns out that carbon dioxide, which is released from respiring tissues, remember carbon dioxide is a waste product of cellular respiration, lowers the pH in those tissues, because remember carbon dioxide and water together make carbonic acid, and that releases protons, which lowers the pH. And a lower pH actually changes the shape of the hemoglobin subtly and lowers the stickiness or the affinity of hemoglobin for oxygen. Now, that means that in areas where there is high carbon dioxide concentrations and lower pH, which is happening at respiring very active tissues, well, it turns out those are also the tissues that need more oxygen because they are actively respiring and oxygen is needed for cellular respiration. So what happens is in those tissues that need more oxygen and are releasing more CO2, they end up getting more oxygen because the hemoglobin there is less sticky for oxygen. So oxygen gets picked up at the lungs and then it gets dropped off at the tissues, and that is increased, that effect is increased by the fact that the hemoglobin gets less sticky. To show that, we actually have to draw a second curve. In red here, I'm drawing the hemoglobin oxygen dissociation curve at higher CO2 and lower pH. So you can imagine if you wanted to trace the curves around, instead of picking up oxygen at the lungs here and dropping it off at the tissue here, you actually end up dropping it off at the tissue here on this part of the curve, meaning we're at a lower saturation, and that gap, instead of being this gap here, is greater. So again, at higher CO2, and lower pH, hemoglobin is less sticky, and that's reflected in this rightward curve. This is called the Bohr effect, or the Bohr shift, because we talk about the graph being shifted to the right, although I find it easier to think of the graph being shifted down as we move from blue to red. Now, before we finish, there's one more form of hemoglobin we need to talk about, and that is fetal hemoglobin. Fetal hemoglobin is the hemoglobin that fetuses have in utero, and it has a different structure. Instead of 2 alpha and 2 beta chains, it has 2 alpha and 2 gamma chains, and that results in it being stickier for oxygen. Fetal hemoglobin has a greater affinity for oxygen at all partial pressures, and so its hemoglobin or oxygen dissociation curve looks like this. It's shifted to the left from the adult hemoglobin, and so at any partial pressure, it has a higher oxygen saturation. That's what the graph being leftwards or upwards of the blue graph is showing us, that it is stickier for oxygen. And that's important because fetal hemoglobin's greater affinity for oxygen allows it to steal or take oxygen off of the maternal hemoglobin. Maternal hemoglobin in maternal blood comes close to fetal hemoglobin in fetal blood. They don't actually mix, but they come close together in the placenta, and the fetal hemoglobin steals the oxygen off the maternal hemoglobin, allowing the fetal circulation to provide the fetus with oxygen. It needs to be stickier for oxygen in order to be able to get oxygen from the mom. So that is a look at hemoglobin and oxygen dissociation curves, including the Bohr effect and fetal hemoglobin.